It's important to note that enzyme activity is regulated, meaning that enzymes are not always active all the time. And that's important because the metabolic needs of cells change over time. So, for example, during development, if we look at the kind of the developmental time known as puberty, we know that there are certain cells that become more active, and those cells start secreting hormones that are involved in the kind of the process of puberty. And so these were enzymes that were maybe turned off or not active earlier in life, but then they become activated during that time known as puberty. Different cells in the body also have different metabolic needs. So some cells need to have some activated enzymes, whereas other cells might not need those same enzymes to be activated. And so there has to be a way in order to turn on enzymes when they're needed and turn them off when they're not needed. In the rest of this video, we're going to talk about these different ways that enzymes are regulated. So we're going to talk about competitive inhibition, non-competitive inhibition, which also includes a subtype called allosteric inhibition. We will take a look at allosteric activation, which includes things called cofactors and coenzymes. We'll talk a little bit about feedback inhibition, and then we'll touch upon the fact that enzymes are compartmentalized in different organelles within the cell. So let's start by taking a look at competitive inhibition. So competitive inhibition is when a molecule other than the substrate is able to bind to the active site. And when that molecule binds to the active site of the enzyme, it blocks the substrate from binding to the active site, and therefore the enzyme is not able to catalyze the reaction. So this top reaction that we see up here, this is the normal reaction. And you see we have the enzyme here as this kind of gray Pac-Man looking thing. We have our substrate up here. And when the substrate binds to the active site, it causes the substrate to break into two pieces. So basically the substrate is actually made up of these two pieces here that get separated by the enzyme. Now, in competitive inhibition, that's what we're seeing down here at the bottom, where we have something that looks kind of like the substrate. So it still has a similar shape as the substrate, but it's not the substrate. And what happens is that it binds to the active site and it physically blocks the active site. So now the substrate can't get to where it needs to go, which means that the enzyme can no longer catalyze that reaction because the substrate can't get to the active site. Non-competitive inhibition has the same outcome as competitive inhibition in that the enzyme is not able to access the substrate to catalyze the reaction. However, it's accomplished in a slightly different way. So non-competitive inhibition occurs when a molecule other than the substrate binds to an enzyme at a location that's not the active site, but it still blocks the substrate from binding to the active site. So when we say a molecule other than the substrate, this would be our inhibitor. And it's going to bind to the enzyme. It's just going to bind to it in a spot that is not the active site. So once again, in this picture down here, the top one is showing us a normal reaction where we have the substrate fitting into the active site of the enzyme, and the enzyme catalyzes the reaction to break that substrate down into its two pieces. In the bottom one, we see the inhibitor. In this case, the inhibitor is not binding to the active site, but it's binding to a secondary site. But the result is the same because, as you can see here, it's now blocking the active site. So the substrate can't get to the active site, which means that the enzyme cannot break down the substrate. Allosteric inhibition once again has the same outcome as competitive and non-competitive inhibition. The outcome is that the enzyme cannot catalyze the reaction. The way that it works is just slightly different. Allosteric inhibition is actually a type of non-competitive inhibition because the inhibitor binds to a spot that is not the active site. So allosteric inhibition is when a molecule binds to a site other than the active site and changes the conformation. And what we mean by conformation is that it's changing the shape of the active site. And if you change the shape of the active site, the substrate can no longer fit into the active site like a lock and key. So once again, on this picture, the top reaction is the normal reaction. And the bottom reaction is showing us the inhibition. So in this case, the inhibitor binds to a secondary site known as the allosteric site. So it's not binding directly to the active site, it's binding to a different part of the enzyme. But remember that enzymes are proteins and they have this three-dimensional shape. And so if something binds to one part of the protein, it can actually kind of cause the rest of the protein to change a little bit in its shape. And you'll notice that the active site here, which was kind of pointy, has now been changed by that inhibitor. And that change in shape prevents the substrate from being able to bind to the active site. And so once again, the outcome is the same. The outcome is that the substrate and the active site of the enzyme can no longer interact and the reaction cannot be catalyzed. In addition to inhibiting an enzyme's activity, there may also be a requirement to activate an enzyme. And so we actually have these kind of helper molecules called cofactors and coenzymes. We'll talk a little bit more about these specifically on the next page. But essentially, these molecules can bind to an enzyme on a spot that's not the active site. So it actually binds to the allosteric site. Now, previously, when we talked about an inhibitor binding to the allosteric site, it changed the shape of the active site so that the substrate could no longer fit and the enzyme could not catalyze the reaction. But in allosteric activation, what happens is that these molecules bind to the allosteric site and it still changes the conformation. And remember, the conformation is just the shape of the active site. But in this case, it actually changes it to a shape that makes it fit the substrate. So it's actually making the active site fit the substrate. So we can see in this picture here that our substrate is this kind of half circle looking molecule here. And so if we were to draw that up here, you can see that the substrate, it's not able to fit into that active site because it's not the right shape. But if we have this activator, which is going to be our cofactor or our coenzyme, when it binds to the allosteric site, which is that region right there, you'll notice that it changes the active site from this kind of V shape to this rounded shape. And this rounded shape allows the substrate to bind with the active site. And now that substrate can be catalyzed. The reaction can occur. So let's talk a little bit more about those cofactors and those coenzymes. And remember, these are the allosteric activators. And these are molecules that are not proteins, but they help to bind to the enzyme and they optimize the conformation. Remember, that's the shape of the active sites. And so the main difference between a cofactor and a coenzyme is just that a cofactor is inorganic and coenzymes are organic. And generally speaking, the cofactors are going to be the minerals that are required in your diet, whereas coenzymes are going to be the vitamins. And this is why it's important for you to get the right amount of the right kinds of vitamins and minerals in your diet because you have to have the correct cofactors and coenzymes to help activate your enzymes to make them work properly. Another form of enzyme inhibition that I would like to talk to you about is feedback inhibition. And feedback inhibition is kind of like a self-regulating reaction. So remember that a reaction can be written as a reactant, and then an arrow, and then if there's an enzyme involved, you write the name of the enzyme above the arrow, and then that gives you the product. Remember, the reactant is also known as the substrate. Well, in feedback inhibition, what happens is that the product becomes the inhibitor. So you can see in this picture at the bottom that we have a substrate that binds with an enzyme, and that produces a product, which in this case, that product actually becomes the next substrate. So this is like substrate number 2. And that substrate binds with a second enzyme. So we'll call this one enzyme 1 and this one enzyme 2. And when that substrate binds with that enzyme, it gives us this product, which then becomes the next intermediate substrate. So this is like our third substrate. And when that substrate binds with our third enzyme, it produces this product here. And this product ultimately becomes the inhibitor. And so it can actually bind to the original enzyme 1 and kind of shut it down. So this would be an example of like an allosteric inhibitor because it changes the shape of the active site. But the key thing, regardless of whether it is an allosteric or another form of non-competitive inhibition, the key thing is just that the product becomes the inhibitor. And so what this means is that as more and more product is being made, more and more of the enzymes are going to be kind of turned off or down-regulated. And so that's why I said this is kind of a self-regulating reaction. It's because the product actually shuts down the reaction when it starts to get too concentrated. And then last but not least, I would like to point out that enzymes are also compartmentalized both within cells and within the body. So we've talked quite a bit, especially about these eukaryotic cells that have these different membrane-bound organelles. And so these different organelles are specialized for what they do. And so only the enzymes that are needed in the particular organelles are going to be made because it takes energy and materials for a cell to make these enzymes. So why would it make enzymes for an area where it doesn't need them? So some examples are things like DNA polymerase. Remember that ASE, ending in ASE tells us that this is an enzyme. And this is specifically an enzyme that makes a polymer of DNA. Well, DNA polymerase is only located in the nucleus because that's where DNA is located. There's another enzyme that we're going to talk about in an upcoming video called ATP synthase. And once again, ASE tells us it's an enzyme, and specifically, it's an enzyme involved in the production of ATP. And it's only found in the mitochondria because that is where ATP is primarily produced. Now, you would not find ATP synthase in the nucleus, and you would not find DNA polymerase in the mitochondria because the other materials needed for those processes are not located in those other organelles. Each organelle is highly specialized as to what it does. In addition to individual cells, individual parts of an organism also make different enzymes. And that's because different parts of the body need different enzymes to carry out their function. For example, in the human body, the enzymes needed to make hormones are going to be found primarily in the endocrine glands. And the enzymes needed for digestion are going to be found in the digestive system. And so once again, the body is not going to make enzymes that it doesn't need because that's a waste of energy and materials.